What if a single drop of blood could tell you that you were going to get cancer years from now? Today, we're going to trace the decade-long scientific journey that turned that incredible idea into a reality. I mean, it sounds like something straight out of a sci-fi movie, doesn't it? But here's the thing. It's not. This is real, and it's the culmination of a series of brilliant breakthroughs that all started with a really fundamental question. So let's dive in. Back in 2013, a group of researchers started this incredible hunt, a hunt for what you could call cancer's ghost, hiding right there in the bloodstream. We now know this concept as a liquid biopsy. And the challenge here? Well, it's absolutely immense. Picture this. You're in a massive, noisy ballroom, and you're trying to hear a single spy whispering in the corner. That whisper? That's the tumor DNA. And the noisy crowd? That's all the DNA from your healthy cells. It's an almost impossible task. So, in the early days, the strategy wasn't to look for one specific kind of cancer. Instead, they were looking for these broad, genome-wide signals, kind of like a general alarm that goes off no matter which cancer it is. And right from the get-go, they hit this really interesting trade-off. On one hand, you had hypomethylation analysis. It was way more cost-effective, and it worked pretty well even with a small amount of data. On the other hand, you had CNA analysis. This one could look for more specific changes, but it needed a ton more data to be accurate. So, it's that classic dilemma. Do you go for the cheaper, broader tool, or the more expensive, precise one? But you know what? Those early results were incredibly promising. I mean, they were able to detect several different cancers: liver, breast, lung. And when they combined both methods, the accuracy shot up. This was huge. It proved the whole concept could work. The ghost was real. But to really prove its value, to turn this from a cool lab experiment into a real-world tool, they needed to narrow their focus. And that brings us to our next chapter. To really prove this thing could save lives, they needed a high-stakes test case. They needed to pick a fight with a very specific kind of cancer. So they zeroed in on something called nasopharyngeal carcinoma, or NPC. Now, what's so special about this cancer? Well, it's strongly linked to the Epstein-Barr virus, EBV. And here's the genius part. The EBV DNA in the blood acts like a giant flashing neon sign that screams cancer is here. It made it the absolute perfect target for a blood-based screening program. So in 2017, they went all in. They launched this massive screening study. We're talking over 20,000 men in a high-risk area, none of whom had any symptoms. The goal was simple but profound. Could this blood test for EBV DNA find NPC early enough to really matter? And look at these results. I mean, wow. It was a dramatic success. The screening completely flipped the script. In the past, only about 20% of these cancers were caught at an early stage. With this new blood test, that number skyrocketed to 71%. It was a total game changer, catching the disease when it's most treatable. I mean, this was a life-saving breakthrough, no question about it. But, and there's always a but in science, right? This incredible success actually created a whole new, unexpected problem. So what was the problem? Well, it turns out only 11% of the people who got a positive result actually had cancer. That's a really low positive predictive value. It meant a ton of false positives, which causes so much unnecessary anxiety, not to mention more invasive follow-up procedures. The tool was powerful, for sure, but it wasn't nearly precise enough. Okay, so to tackle that false positive problem, the team knew they had to get smarter. They couldn't just look for the presence of the DNA anymore. They needed to go deeper. They had to figure out what the cancer DNA looked like. And what they came up with in 2019 was this brilliant three-signal approach. It wasn't just about if the EBV DNA was there. It was about analyzing its specific characteristics. First, the quantity. How much is there? Second, the size. What's the length of these DNA fragments? And third, methylation. What's its epigenetic pattern? It's like building a much more detailed suspect profile. And this was the key. They discovered that the EBV DNA coming from a tumor was totally different from the background viral DNA. It was more plentiful, sure, but it also had a really specific size profile and a unique methylation pattern. Suddenly, that whisper in the ballroom didn't just exist. It had a distinct accent they could pick out from the crowd. And by combining all three of these signals, bam, they more than tripled the accuracy of the test. The positive predictive value jumped from 11% to over 35%. That meant drastically fewer false positives. It was a massive leap forward. And this whole idea,
looking at the characteristics of the DNA fragments, not just their existence, well, that was the beginning of a whole new, even more advanced field, fragmentomics.
So what is fragmentomics? Well, the idea is pretty cool. It's the study of the patterns of all this circulating DNA. Think about it. When a cell dies and its DNA breaks apart, it doesn't just shatter into random bits. The way it breaks, the size of the pieces, even the little genetic sequences at the very ends of the fragments, all of it leaves behind clues about where that DNA came from, a healthy cell or a cancer cell. So these researchers started digging into all of it. They looked at the size profile of the fragments. They analyzed the end motifs. That's the specific DNA letters at the very tip of each fragment, which can tell you which enzyme cut it. They even developed a technique called fragma to figure out methylation patterns without needing any chemical treatments. This gave them an arsenal of tools to see even the faintest, most specific signals of cancer. And this is what set the stage for the final, absolutely mind-blowing discovery. Okay, so this is where the story goes from incredible science to something that feels like science fiction. Years after that initial 2017 screening, the researchers did something critical. They followed up with the original participants, and what they discovered in those old blood samples, well, it completely shifted the paradigm. Here it is. They found that the people who eventually developed cancer years later already had these distinct fragmentomic signatures in their blood all the way back in that first test. Let that sink in. The blood test wasn't just a snapshot of the present. It was a crystal ball. It was identifying a molecular state that came before the clinical appearance of the disease. It was predicting the future. And the risk factor here is just staggering. People with the full set of these high-risk molecular features in their blood had an 87 times greater risk of being diagnosed with cancer down the road compared to those who tested negative. 87 times! This wasn't just detection anymore. This was prediction. So when you look back at this whole decade of discovery, you can see how each step built on the last. It starts in 2013 with just a concept, proving you can even find these signals. Then, 2017, they put it to the test in a massive screening, showing it can find cancer early. 2019, they refine it, making it way more accurate. And that all leads to 2025, where they realized they didn't just have a detection tool, they had a predictive one. And this whole journey, this incredible story, it's really a blueprint. Remember, this was all for just one type of cancer. So it leaves us with this huge, tantalizing question. What's next? If we can do this for one cancer, what could this new paradigm of prediction mean for the future of medicine as a whole?